In the last video, we learned the rules of heredity, Mendel's laws. We emphasized the distribution of alleles during gamete formation and their pairing during fertilization. If you think carefully about it, you may realize that the distribution of alleles in gametes and their combination upon fertilization are random. Because they're random, they're governed by simple laws of probability. So let's take a few minutes now and learn about probabilities. The probability that an event will happen is a number between 0 and 1. The probability of an event that is certain to happen is 1. The sun will rise tomorrow has a probability of 1. The probability of an impossible event is 0. The probability that you will pass general biology is 0. Kidding. Maybe this is a better example. The probability that Earth will suddenly lose its gravity and will all float off into space is 0. Between these extremes, all other probabilities are some value between 0 and 1. Let's look at an example. Let's say you have a baby. The probability that it's a girl is 50%, or P girl equals 0.5. It's actually not exactly 0.5, but it's very close. The probability that your baby is either a boy or a girl is 1. There are no other possibilities. The probability that your child is neither a girl nor a boy is 0. Those are the only two choices. Often in genetics, as in other applications of probability, we'll want to know how likely it is that two events happen together. In these cases, we apply the product rule, which states that the probability of two events is equal to the product, that means you multiply, of their individual probabilities. Whenever two events are connected by the word "and," you multiply their probabilities. The probability of A and B is equal to the probability of A times the probability of B. Here's an example. Say you're headed to the hospital to deliver a baby, and a friend of yours is having a baby at the same time. What are the odds that you and your friend both have a girl? Note the use of the word "and." The probability is the product of the possibility of each of you having a girl. That's 0.5 for you times 0.5 for your friend, or 0.25, or 25%, or 1/4. The other rule in probability that you should be aware of is the addition rule. The probability of an event that can occur in two or more different ways equals to the sum of their individual probabilities. So when events are connected by the word "and," you multiply the individual probabilities. When events are connected by the word "or," you add the probabilities. Let's go through an example. If you have two children, what is the probability that exactly one of those children is a girl? Well, there are two ways that can happen. Either your first child is a girl and your second child is a boy, or your first child is a boy and your second child is a girl. So we calculate the odds of each of those scenarios and add them together. The odds of having a girl and then a boy is 1/2 times 1/2, or 1/4. Likewise, the probability of having a boy and then a girl is also 1/4. Add up those two different ways of having exactly one girl and you'll get 1/2. Now that we know a little bit about probability, let's apply it to the dihybrid cross that we examined in the last video. Remember that Mendel's second law says that traits sort independent of one another. So we can model the inheritance of these traits using the simple rules of probability that we just learned. Here, instead of the 4x4 Punnett square that we used before, we'll use two 2x2 Punnett squares, one for seed color and one for seed shape. On the left, we see the heterozygote by heterozygote cross for seed color. And it shows that 3/4 of the F2s are yellow and 1/4 are green. Likewise, 3/4 of the F2s are round and 1/4 wrinkled, as shown on the right. Since this is a dihybrid cross, we want to know how likely is each combination of phenotypes, round and yellow, round and green, etc. Since we're combining things with the word "and," we use the product rule. The probability that a seed is round and yellow is equal to the probability that it is yellow times the probability that it is round, or 3/4 times 3/4, or 9/16. The probability that a seed is yellow and wrinkled equals 3/4 times 1/4, or 3/16. The probability that a seed is green and round is also 3/16. Finally, the probability that a seed is green and wrinkled equals 1/4 times 1/4, or 1/16. Note that we've reproduced the 9 to 3 to 3 to 1 ratio that we saw earlier, this time using simple arithmetic rather than counting squares in a 4x4 Punnett square. Which approach you use in a situation like this is up to you. For me, it's easier to do the multiplication, and I never draw anything larger than a 2x2 Punnett square. For you, you may choose to do the 4x4 Punnett square when following two traits. When following 3 or more traits, the Punnett squares become too unwieldy and you have no choice but to figure it out arithmetically. Let's put together what we've learned about probabilities, Punnett squares, and crosses and use them for a little detective work, the test cross, which allows you to figure out an unknown genotype. For example, you know the genotype of a plant with a recessive phenotype. It's homozygous for the recessive allele. It's the only way you can get the recessive phenotype, white flowers in this case. If, however, your plant has the dominant phenotype, purple flowers, then you don't know its genotype. It could be either homozygous or heterozygous. You can distinguish these possibilities with a test cross. In a test cross, an individual of unknown genotype is mated with an individual of known genotype. Once again, you know the genotype when you see the recessive phenotype. So the white-flowered plant serves as the mating partner of known genotype. If 100% of the offspring have the dominant phenotype, then the genotype of the parent is homozygous dominant. If the offspring are 50% dominant phenotype and 50% recessive phenotype, then the parent is heterozygous. Those are the two possible outcomes of a test cross, either 100% dominant or 50-50. Mendel's laws faithfully describe the segregation of alleles in all situations, but there are a few extensions of the laws that you should be aware of: incomplete dominance and codominance. There are other modifications as well, and we'll cover those later. For now, we're going to cover these two, and we'll start with incomplete dominance, which is shown here. I'll try and stick to the term incomplete dominance, but sometimes you'll hear the terms semi-dominance or partial dominance. The definition is given here. In incomplete dominance, the phenotype of the heterozygote is intermediate between the phenotypes of the homozygotes. I'll go through some examples that hopefully make this clear, but if you have trouble with it, at least memorize this definition. An example is shown here, with red and white flowers. To avoid any confusion, let me say that these are not pea plants. With pea plants, we talked about purple versus white flowers, and purple showed complete dominance over white. This is a different situation in a different species of flowering plant. At left, you see a cross between a red-flowered plant and a white-flowered plant, with each of the parents being true breeding, which means they're homozygous for their respective alleles. Capital R for red and lowercase r for white. We've seen this before. When two plants that are homozygous for their traits are mated, all of the offspring are heterozygous. That's just what happens here. But those heterozygous plants have pink flowers, pink being intermediate between red and white. That's the definition of incomplete dominance. The heterozygote has an intermediate phenotype. Let's take this a little bit further and look at the situation on the right. Here we have a cross between two pink flowers, which are both heterozygous. This is the typical heterozygote by heterozygote F2 cross that we've seen before. As we've seen before, it yields offspring in the ratio of 1 homozygous red to 2 heterozygous to 1 homozygous white. In this case, the phenotypic ratio is not 3 to 1 as we've seen before. Rather, it matches the 1 to 2 to 1 genotypic ratio because the heterozygotes have their own phenotype. Note also that we know the genotype of the plant just by looking at its flowers. No test cross is necessary. Now let's look at another real-world example of incomplete dominance. This one relevant to human health. Familial hypercholesterolemia is a genetic disease that leads to abnormally high cholesterol levels in the blood. I know that's a mouthful, so just say FH for short. Normally, the liver removes cholesterol from the blood, and that helps keep blood cholesterol within a normal range. In FH, a protein on the surface of liver cells no longer works properly, and the liver does not remove cholesterol from the blood as it should. The result is high blood cholesterol, which is dangerous in that it makes a heart attack more likely. It is a rare disease, and the milder form only affects about 1 in 500 people. This table shows the cholesterol level and tendency to heart attack of people based on their FH genotype. Plus represents people with the normal genotype and no disease. Their blood cholesterol is not necessarily elevated and stays in a normal range, and they have the typical risk of heart attack, which comes later in life, usually past the age of 60. FH is the disease allele, and plus FH individuals are heterozygous for the disease allele. They have elevated blood cholesterol, about twice normal. They are also prone to heart attack much earlier in life, heart attacks happening in the 30 to 50 age range. FH individuals are homozygous for the disease allele, and they have greatly elevated blood cholesterol, 6 times normal. They are also prone to heart attack at a very early age, often before age 10. This is incomplete dominance since heterozygotes have a phenotype intermediate between the two homozygotes. Of course, there's another way of looking at it. If you consider just the more severe form of the disease, which is devastating, you could say that it's simply recessive. The second modification to Mendel's laws is codominance, in which both alleles or both traits are expressed. This is distinct from incomplete dominance, so keep the different definitions in mind. The quintessential example of codominance is the ABO blood group, which you're probably familiar with. Your blood type, A positive, O negative, etc., determines who can accept blood that you donate or who can donate blood to you. One blood type may or may not be compatible with another because of the antigens on the surface of red blood cells. An antigen is something that the immune system sees and sometimes attacks. As you can see in the illustration, red blood cells have different antigens on their surface. Someone with type A blood has the A antigen on the surface of their red blood cells, denoted here by pink circles. Type B has the B antigen, denoted by green diamonds. But AB blood is also possible with red blood cells that have both the A and B antigen. This is codominance. Both traits are present, type A and type B blood. As you can see, it's also possible to have neither type A nor type B blood. Such people are type O and have neither type of antigen on their red blood cells. Now let's look at the genetics of this situation. A single gene determines the ABO blood type. There's also a separate gene that controls positive versus negative blood, but we're not talking about that right now. We'll take a closer look at that when we do the genetics lab. For now, let's confine ourselves to the ABO gene. So far, all the genes we've seen have 2 alleles, purple versus white flowers, brown versus wrinkled, seed shape, etc. This situation is different because there are 3 alleles. There's nothing magic about 2 alleles. If we look across many people, we're not limited to just 2 alleles. In the case of the ABO blood type, there are 3 alleles. 2 that are dominant, hence codominance, and 1 that is recessive. The allele that encodes type A blood is denoted I superscript A, and the allele that encodes type B blood is I superscript B. Capital I in both cases because they're both dominant, and A is differentiated from B by the superscript. The recessive allele, which encodes O blood, no antigen, is denoted by the lowercase I. So the genotype of someone with type A blood is either homozygous IA or heterozygous IA and the recessive allele. Likewise, someone with type B blood is either homozygous IB or heterozygous IB and the recessive allele. If, however, you're heterozygous and have both dominant alleles, IA and IB, then your blood type is AB. Once again, codominant because both alleles are expressed. Finally, you have type O blood if you have both recessive alleles.